The study I'm going to present today is genetic mapping for stripe rust resistance in the hexaploid spring wheat population. Before presenting my actual research work, I would like to give a brief description about wheat production in Canada and all over the world. Wheat is a significant food for human nutrition and consumed by billions of people all over the world. And this is the reason that wheat crop is being widely cultivated worldwide. In 2019, approximately 216 million hectares of wheat was harvested globally, with the total estimated production of 766 million tons. And Canada is one of the top 5 wheat producing nations in terms of acreage and annual production. In 2019, approximately 9.7 million hectares of wheat was harvested in Canada, with the total production of 32.3 million tons. But wheat production in Canada is threatened by several biotic and abiotic factors. Stripe rust caused by Paccinia stripiformis is one of the most devastating fungal diseases of wheat in Canada. In this disease, we observed yellow to orange colored pustules in vertical lines that run parallel to leaf veins. It can adversely affect wheat yield and quality. This disease has always been a problem in southern Alberta, but now it has also spread in other parts of Canada. In Western Canada, several epidemics have been reported in 2005, 2006, 11, and 16. There are two main reasons for this recent expansion of stripe rust severity in Western Canada. Firstly, this pathogen was limited to cooler region in the past, but now new races have evolved which are adapted to higher temperature. And secondly, this pathogen has been overwintering on winter wheat. So to overcome this situation, we need to identify new sources of stripe rust resistance. We have unknown sources of resistance in our local Canadian cultivar, which need to be characterized. For example, AAC ANOVA is a Canadian spring wheat cultivar, and it possesses effective level of stripe rust resistance in field condition, but its genetic resistance was still unknown. So the objective of my study was to identify and locate QTL controlling genetics of stripe rust resistance in AAC ANOVA. To achieve this goal, I followed all these steps. First of all, we developed our double haploid mapping population, and then we planted that population at 2 sites to collect disease-weighted data. And then I analyzed that disease-weighted data followed by DNA extraction and genotyping. And then I constructed a linkage map and utilized that linkage map and disease-weighted data for QTL analysis. In next slides, I will explain each step in detail. So in the first step, we developed a double haploid mapping population comprising of 291 lines. And this population was generated from the cross of a susceptible cultivar AAC Proclaim with a resistant cultivar AAC Innova. And we use wheat maize hybridization method for the production of this double haploid mapping population. And then we planted that double haploid mapping population at 2 sites, Creston, British Columbia, and Lethbridge, Alberta. Disease pressure was very high at both sites because they are very close to the Pacific Northwest of United States, where original epidemics occur and then rust spores travel towards Western Canada. There was a very little chance for any plant to escape from the disease at both locations, and it gave us very good opportunity to evaluate that population response under natural infection. We planted our experiment in randomized complete block design, and then we recorded disease-weighted data 2 to 3 times visually on modified Cobb scale, where 0% was immune and 100% was completely susceptible. And then I analyzed that data using our software, and here is a ANOVA table for disease severity of double haploid mapping population. And you can see the effect of genotype environment and genotype by environment interaction was highly significant on stripe rust severity. And this is the histogram showing frequency distribution of stripe severity among double haploid lines across all 5 environments. And first arrow is showing the mean value of stripe severity for resistant parent AAC ANOVA, and that mean value was around 4%. And second arrow is showing mean value of stripe severity for susceptible parent AAC proclaim, and that mean value was 73%. And this histogram is showing continuous distribution of stripe severity among all double haploid lines and skewed towards greater resistance. And in the next step, I extracted DNA from all double haploid lines and parents. And for the extraction of DNA, I took fresh leaf samples from 2 weeks old seedling. And then I used this commercial kit for the extraction of DNA. And then I dissolved DNA pellet from each sample into 100 microliter of 1xTE buffer. In the next step, I quantified each sample individually using this commercial quantification kit, and then I diluted each sample to 50 nanogram per microliter. After extraction of DNA, I sent DNA samples from parents and 190 double haploid lines for 90K Infineum X-Lips assay. And we received a total of 587 SNPs from provider. And then I performed these filtration steps. First of all, I used Genome Studio software for the filtration of high quality SNPs. And I removed all monomorphic markers from the analysis, and markers with more than 10% of the missing values were also excluded from the analysis. And then I performed cascade test and removed markers that were significantly distorted from the expected segregation ratio. After performing all these filtration steps, a total of 7,369 SNPs, which was approximately 9% of the original data set, was used for the construction of linkage map. And then I constructed a linkage map by using these two software, MST Map and Map Disto. First of all, I created draft linkage group in MST Map software, and then I refined each linkage group individually by recalculating marker distance and order within the linkage group. And here are the results for linkage map created for this double haploid mapping population. In this map, a total of 7,112 markers were incorporated into 29 linkage groups on all 21 read chromosomes, and the total map length of 29 linkage group was 3,074 centimorgan, and average distance between two markers was 2.31 centimorgan. Since many markers segregated together, the number of linkage events for this map were 1,381. And if we consider read subgenomes, then highest number of markers were mapped on B genome, followed by A genome and D genome respectively. And then I utilized that linkage map and disease weighted data for QTL analysis. And I performed this analysis in Windows QTL cartographer software with the window size set at 1 centimorgan, walk speed of 1 centimorgan. And I also performed 1,000 permutation tests to determine significant load threshold. And this QTL analysis was undertaken for each environment individually and across the environment using pooled data. And here are the results for this QTL analysis. In this study, we identified 11 QTLs, 1 major QTL, and 10 minor QTLs. Major QTL was identified on chromosome 2A, and it explained 14 to 33% of the phenotypic variance among all 5 environments. And this QTL was tracked from our resistant parent AAC ANOVA. And then 4 QTLs were identified on chromosome 2B. They all were minor QTL because they explain less than 10% of the phenotypic variance and tracked from our resistant parent AAC ANOVA. And interestingly, our susceptible parent AAC proclaim contributed alleles for QTL on chromosome 2D, 5D, and 7D, but they all were minor QTL and explain less than 10% of the phenotypic variance. The second most stable QTL in present study was identified on chromosome 5A, but it was a minor effect QTL, which explained 5 to 7% of the phenotypic variance. Similarly, QTL identified on chromosome 3B and 5B were also minor effect QTL and derived from our resistant parent AAC ANOVA. So the most significant QTL in present study was identified on chromosome 2A. And here is the load graph for this QTL. And this QTL was mapped on the distal end of chromosome 2A within 218 to 240 centimorgan interval. And load value for this QTL varied from 8 to 21 across all 5 environments. These horizontal lines are indicating the load threshold determined after 1000 permutation tests, and these load threshold varied from 3.2 to 3.4 across all 5 environments. And 3 most stable QTL identified in this study on chromosome 5A, 7D, and 2A were also assessed individually and in combination for their effect on stripe severity. And you can see the major QTL identified on chromosome 2A was the only QTL responsible for the reduction of stripe severity individually, while QTL identified on chromosome 7D and 5A were only effective in combination. And although we identified 11 QTL in this study, but QTL identified on chromosome 2A was the most important QTL in this study. We can utilize this QTL in further breeding program, but before utilizing it into marker assisted selection, we need to convert its closely associated marker into test markers. In future, we will incorporate that QTL into other commercial Canadian spring wheat cultivars and paramit with other genes for stripe resistance. And at the end, I would like to acknowledge my supervisor, Dean Spanner, co-supervisor Harpinder Randhawa, and committee member Brian Berry. And we highly acknowledge financial support provided by Alberta Wheat Commission and Saskatchewan Wheat Development Commission for their financial support through project grants. And I'm also highly thankful to Alberta Wheat Commission and Western Grain Research Foundation for giving me graduate research scholarship to pursue my MSc at University of Alberta. Thank you.